Today we will start chapter number 20, that is locomotion and movement. Locomotion, the literal meaning of locomotion is that when the body or the organism move and change the place, then it will be known as locomotion. Whereas the movement occurs at that same place but place is not changed, site is not changed, that it will be known as movement. So movement is a part of locomotion, and when movement occurs, then along with the place change, then it will be known as locomotion. Now in this chapter we'll start with the muscles. Muscles are of three types. We have already discussed in the previous classes that these muscles are of three types which help our body for various activities. These are a skeletal muscle, which is also known as striated muscle due to its appearance, that is light and dark band. Then the smooth muscles, they are devoid of any striation, that's why known as smooth muscles, and cardiac muscles. Now we will discuss about 3 points of all these muscles, that is structure, location and function. Skeletal muscle, as the name suggests, it is attached with our skeletal system. Then smooth muscle present in the visceral organs, internal organs. Cardiac muscle present in our heart. Now this skeletal muscle present along with attached with the skeletal system. It is long thread-like structure and having dark and light striations. Then it is not branched, as well as they helps in the process of movement, locomotion, etc. Whereas smooth muscles are just like the such type of structure without any striation, and that's why it is known as smooth muscles. No striations are there. They are present in the visceral organs, internal organs. They are involuntary in function. They are not working according to our will, whereas skeletal muscles are voluntary in nature because they work according to our will. Cardiac muscle present in the heart, and they are also having the dark and light striations, as well as they are branched. They helps in the movement of heart, that is pumping of heart. They are myogenic in nature. They are also involuntary in nature. That we will discuss through the diagram. Then next topic is the structure of contractile protein. These skeletal muscles are the structure of now in this chapter we will discuss about these muscles, that is skeletal muscle, because skeletal muscles are helping in the process of movement and locomotion. This contains various muscle fibers which are in a group, and that group, the bundle, is known as the fascicles. These fascicles having numerous muscle fibers. Now when we'll see the structure of muscle fiber, we can see the plasma membrane of the muscle fiber is known as sarcolemma, whereas the protoplasm is known as sarcoplasm. Then the endoplasmic reticulum is known as sarcoplasmic reticulum, which is the storehouse of the calcium ion, which will help during the skeletal movement, sliding movement. These muscle fibers have 2 types of protein, that is actin protein and myosin protein. Due to that, it appears dark and light band. These 2 types of contractile proteins helps in the process of the movement. The actin protein is also known as thin F protein or filamentous protein, and it consists of 2 filamentous helically wounded structure. Then these two filaments are also having the tropomyosin, and these tropomyosin are close to the F actin, that is this filamentous actin. These filamentous actins are monomeric actins of globular protein, and along the whole length these tropomyosin run parallelly. At regular interval, troponin is present on this tropomyosin. During the resting phase, this tropomyosin masks the other type of contractile protein, that is myosin. So this is the next type of myosin. The second type of contractile protein is myosin, which is also known as thick filament. This consists of meromyosin, numerous meromyosin unit, which has two parts, that is head with short arm and tail with the tail. This head is also known as HMM, and that is heavy meromyosin, and the tail is also known as LMM, that is light meromyosin. This head part is active and having the active ATPase, and it has two sides for binding of ATP as well as binding for actin protein, which will help during the movement or the sliding movement. Now that we will discuss in the diagram. And we will also discuss the mechanism of muscle contraction, how the muscle contract, what are the changes takes place, what are the important phenomena takes place during the mechanism of muscle contraction through this diagram.
In this slide we can see different types of muscles and their functioning, in which part they are present. So there are three types of muscle, that is, first is the skeletal muscle, second is cardiac, and third one is the smooth muscle. The skeletal muscles are attached with the bones, whereas cardiac muscle is present in the heart as well as the internal organs or the visceral organs having this smooth muscles. The shape is also different. In a skeletal muscle it is elongated and cylindrical cell but without any branching, whereas heart muscle is also, cardiac muscle is also such type of structure, elongated structure, but it is branched. And smooth muscles are, the structure is like this, and that's why it is known as what is the structure of the smooth muscle. In this slide we can compare, we can see different types of muscles. There are three types of muscles depending upon their location, shape, functioning, etc. These are skeletal muscle or striated muscle, cardiac muscle, as well as smooth muscle or unstriated muscle. Now the skeletal muscles, as the name suggests, it is attached to the bones and helps in the movement of bone as well as locomotion of the body, whereas heart muscle is present in the heart, and the cardiac muscle helps in the regular rhythm pumping of the heartbeat, all these things. Now smooth muscles are present in the internal organs, that is visceral organs. The shape of this skeletal muscle, they are elongated as well as cylindrical and having more than one nucleus, that's why known as multinucleate. They have dark and light bands which are in alternate way. So these dark and light band, due to this it is known as striated, whereas in a smooth muscle it is spindle shape having one nucleus, that is uninucleate. That means on both the ends it is very narrow pointed, whereas in the middle it is broad. That's why it is known as spindle shaped. It has no dark and light band in the cell, and hence it is known as non-striated. it helps in the movement of the visceral organs, internal organs, and it not work according to our will. That's why it is known as involuntary in nature. Cardiac muscle is also involuntary in nature, whereas this skeletal muscle is voluntary in nature. That means they work according to our will. So in this way we can see there are three types of muscle cells, whereas the skeletal muscle is mainly associated with locomotion and movement process, and that we'll study in detail, already discussed also, that is myofibril and how they helps in the process of movement by sliding movement, sliding theory movement. The structure of two types of filament, that is thin filament made up of the actin protein, whereas myosin, that is thick filament. Thin filament consists the structure of this thin filament we can see here in the diagram. This is the myofibril and how these myofilaments are arranged. This F actin is moving in the whole length, and tropomyosin is also moving along with this in the whole length in such a way, whereas troponin complex, this is the troponin, is present as at regular intervals. Two types of proteins, that is F actin and G actin are there, two lines, and this thick filament, that is myosin. This myosin has two parts, head and tail. Myosin tails are arranged to point towards the center of the sarcomere, whereas the head points to the side of the myofilament band. And then myosin and actin are helping in the process of movement, that we can study through this sliding filament theory, which helps about the understanding the muscle contraction. This theory was first evolved and given by A.F. Huxley and H.E. Huxley in 1950s. According to this theory, the force of contraction is developed by the cross bridge, which in the overlap region in which these two proteins are overlapping. The active shortening is caused by the movement of cross bridge, which is made which cause one filament to slide over the other. This one filament will slide on the other one. During the muscle contraction, the F filament alone show movement, but the myosin filament remain static. The mechanical movement utilizes the energy which is derived from the breakdown of ATP molecules. In this slide we can see the structure of the thin as well as thick filament. Here it is the two filament, that is thick as well as thin filament, how they are arranged. The thick and thin filament, that is actin and myosin, are having the light band. The light band is having the actin, and it is also known as I band or isotropic band, whereas the dark band is known as the A or anisotropic band, and this contains the myosin. So, actin is for the light band and myosin for the dark band. Both proteins are arranged parallel to each other, and actin filament are thinner than the myosin, that is why it is known as thin filament, and myosin is known as thick filament. In the center of the I filament, that I band, which is the actin, which is elastic fiber, the central position is known as the Z line. So, this is the this line will bisect the thin filament. Now, the thick filament is the A band, and they are this A band, this is the thick filament. A band, the central position is known as M line. Now, A and I bond are arranged alternate to each other in the whole length of the myofibril, and in this way they are arranged in such a way. The functional unit of the contraction is known as the sarcomere. So, this is the sarcomere functional unit of this contraction, which is helping in the process of locomotion movement. The central part of this thick filament, this one, is known as the Z zone. So, this is arranged in such a way. Now, the sliding theory, filament theory for the contraction. The myofiber together with all its myofibrils shorten, that is decrease in length by the movement of the insertion towards the origin of the muscle. The shortening of the myofibrils is caused by shortening of the sarcomeres. This sarcomere will decrease in length and the distance between Z line is reduced. So, this distance between Z line will become less, reduced. Now, this shortening of this sarcomere, then the sliding of myofibril will take place and the length of each filament remain the same during the contraction. The sliding of the filament is produced by asynchronous power strokes of myosin of that is given by cross bridge, which is made by this. That means the thin filament, that is actin, over that the thick filament will make a cross bridge like structure. The A band, this one, the A band remain the same in length during the contraction, but are pulled towards the origin of the muscle. Adjacent to this A band are pulled to closer, this portion, and I band between them shorten. The H bands shorten during the contraction as the thin filament on the sides of the sarcomere are pulled towards the middle. So, in this way, by sliding over this one, that is why it is known as sliding filament theory. Filaments are sliding over each other during contraction and as a result the movement takes place. In this slide we can see how the cross bridge are made by this. This is the actin filament and this is myosin filament, thick filament. Myosin head is split ATP and become reoriginated. Now, this myosin head bind to the actin and then it forms the cross bridge. Now, due to the contraction cycle, this ATP is available and Ca, calcium ion level, will become high in the sarcoplasm, that is the plasma, and the myosin head rotate toward center of the sarcomere. All these are aided by the energy ATP. As myosin head bind ATP, the cross bridge detach from actin and it becomes free. Again the cycle will repeat. In this way the movement or the sliding will take place by the help of these two type of muscle, that is protein, that is known as thin filament as well as thick filament. So, the skeletal muscle contraction involves the major steps, that is motor renewal, firing, then depolarization, that is excitation of the muscle cell with the help of the release of calcium ion which is released from the sarcoplasmic reticulum, and shortening of the sarcomere. Now, shortening of the muscle and tension is produced. In this way the muscle contraction will take place, that here we can see in the tabular form also flow chart, that is propagation of the action potential in the molar axon, then the release of acetylcholine that generates action potential in the sarcolemma, that is membrane of the cell, then inward spread of the depolarization along T tubule into the muscle. As a result, the calcium will be released from the sarcoplasmic reticulum and this calcium will bind to the troponin of this thin filament and that which covers uncover the myosin binding site on actin. Now, the myosin head will attach to the actin and the cross bridge cycle will be completed, and due to this the sliding of this thin filament over thick filament toward the center of the sarcomere. All these result in decrease in sarcomeric length, producing muscle shortening, that is muscle will become short and now again it will come in the proper position.